Greetings, class. Have you ever wondered how a fragile, microscopic bubble of water and chemicals survives the chaotic, crushing environment of the microscopic world without bursting or starving? Today, we will study the structural architecture and functional roles of cell membranes.
We have got a packed explainer today. We're breaking this down into 6 parts. 1. Introduction and membrane definition. 2. Phospholipids, barriers and messengers. 3. Cholesterol, the fluidity thermostat. 4. Glycocalyx, receptors and markers. 5. Proteins, transporters, enzymes, scaffolding. And we'll finish up with 6, a high-yield summary and question. Let's get right into it.
Okay, let's dive into Section 1, Introduction and Membrane Definition, the foundation of the cell.
So to truly grasp the brilliance of the cell membrane, we have to clear up a common misconception. The cell membrane isn't just some thin plastic bag holding cellular soup together. It's actually a highly dynamic, complex structural boundary. It's made up of a mix of phospholipids, cholesterol, glycolipids, glycoproteins, and proteins. And the key takeaway here is that each of these components performs very specific, non-negotiable roles to maintain the cell's integrity and regulate its functions. Basically, if you remove even one of these pieces, the entire fortress collapses.
These are the key points related to this topic. Focus on them carefully as they are considered essential for your exams.
Moving right along to Section 2, Phospholipids, Barriers and Messengers, the Structural Foundation. Let's start with the primary building blocks of our microscopic fortress. Phospholipids establish the fundamental structure by forming what we call a bilayer. Now, what's their secret to success? Well, it's all in the tails. Their tails are nonpolar and hydrophobic, which means they literally fear water. This creates an incredible chemical dead zone right in the middle of the membrane, making it super difficult for polar molecules or ions to pass through. It acts like a highly effective microscopic bouncer with a severe water phobia. It blocks unwanted water-soluble substances from entering, while simultaneously ensuring that the cell's precious sugars, amino acids, and proteins cannot leak out.
But here's a wild twist. These structural bricks can actually moonlight as chemical messengers. So some phospholipids can be chemically modified to act as signaling molecules. They can literally move about within the bilayer to activate other molecules, like enzymes. Or alternatively, they can be hydrolyzed, meaning they get split by water, to release small, water-soluble, glycerol-related molecules. These little molecules then zip rapidly through the cytoplasm and bind to specific receptors to trigger internal cellular responses. A great example of this? In your pancreas, this exact system releases calcium ions from the ER, which causes the explosive exocytosis of digestive enzymes. It's just brilliant.
Here are the core details regarding this topic. Review them intently for their deemed crucial for your finals.
All right, Section 3, Cholesterol, the fluidity thermostat, climate control for the cell.
Now, cholesterol gets a pretty bad reputation in diet culture, right? But for your cells, it is an absolute lifesaver. It's a relatively small molecule compared to everything else in the membrane. And just like phospholipids, cholesterol has a hydrophilic head and a hydrophobic tail. Because of this shape, it fits incredibly neatly right between the phospholipid tails. What's really fascinating is how it's distributed. Animal cell surface membranes are packed with it, containing almost as much cholesterol as phospholipid. Plant cells, on the other hand, use it far less, and prokaryotes, they entirely lack cholesterol, relying on very similar off-brand substitute compounds to do the exact same job.
So why do we even need it? Well, it acts as a brilliant dual-action thermostat. At low temperatures, cholesterol prevents those phospholipid tails from packing too closely together, which increases membrane fluidity. This ensures your cells can survive much colder temperatures without freezing rigid and snapping. But at higher temperatures, it stabilizes the membrane so it doesn't just melt into a puddle. Beyond acting as climate control, cholesterol provides essential mechanical stability. Honestly, without it, animal cells would literally burst open. And as a bonus, its hydrophobic regions effectively plug microscopic leaks, preventing ions or polar molecules from escaping. This leak prevention is absolutely critical in the myelin sheath surrounding your nerve cells, ensuring your nerve impulses fire at top speed.
These are the key points related to this topic. Focus on them carefully as they are considered essential for your exams.
Let's shift gears. Section 4, Glycocalyx, Receptors and Markers, the cell's communication network. We've built the wall and set the thermostat, so let's move to the outer surface to see how cells actually communicate. You see, many of the lipid and protein molecules on the outside have short carbohydrate chains attached to them. We call these combination molecules glycolipids and glycoproteins. Picture these chains projecting outward into the watery fluids like microscopic antennae. And they do this amazing magic trick. They form hydrogen bonds with the exterior water molecules, which physically tethers them to the surrounding fluid and helps stabilize the entire membrane structure. Altogether, this sugary cell coating is called the glycocalyx, and brilliantly illustrates how a sugary coating provides actual physical stability. Just remember, in animal cells this is mainly glycoproteins, whereas in plant cells it's primarily glycolipids.
Now these antennae solve a massive problem: cellular blindness. The glycocalyx allows the cell to act as a highly specific receptor network functioning in three main ways. First, you have signaling receptors that recognize messenger molecules like hormones and neurotransmitters. When a messenger binds to the antenna, it triggers a specific domino effect of chemical reactions inside the cell. For example, the glucagon receptor in liver cells responds exclusively to glucagon. Second, you have receptors directly involved in endocytosis. They grab onto structures the cell needs to physically engulf. And third, adhesion receptors physically bind cells together. Without these adhesion receptors, you wouldn't have solid organs. You'd just be a puddle of loose cells.
Building directly on that, the glycocalyx acts as a cellular ID card. Specific glycolipids and glycoproteins function as vital cell markers, or antigens, facilitating cell-to-cell recognition. Think of it this way: each unique type of cell essentially flies its own specific molecular flag, just like different countries flying their own flags. By checking these flags, your immune system can recognize self from non-self in organized tissues safely. A classic, real-world example of this is the ABO blood group system. The differences between blood types are completely based on tiny variations in the carbohydrate portions of these exact surface antigens.
Moving on to Section 5, proteins, transporters, enzymes, scaffolding, the membrane machinery.
We have the barrier, the climate control, and the communication antenna. Now, we introduce the heavy machinery. Because the phospholipid bilayer blocks water-soluble molecules, the cell needs doors. Membrane proteins function as essential transport proteins, providing necessary hydrophilic passageways. These are broadly divided into channel proteins, which act like tiny tunnels, and carrier proteins, which actually change shape to move molecules across. So the crucial point is their strict specificity. Each transport protein acts like an elite VIP bouncer that only lets one exact type of ion or molecule through, ensuring tight control. On top of that, some membrane proteins act locally as enzymes, like the digestive enzymes right on the lining of your small intestine that hydrolyze passing disaccharides. But proteins aren't just for transport. They are foundational to the entire cellular architecture. Some proteins on the internal surface of the membrane physically anchor to a network of internal protein filaments, scientifically known as the cytoskeleton. This internal scaffolding dictates the cell's overall shape, and they're even actively involved in changing that shape when the cell needs to move. Furthermore, proteins play incredibly vital functional roles within the internal membranes of your organelles too. For instance, they are directly involved in running cellular respiration within the mitochondria and powering photosynthesis within chloroplasts.
And finally, Section 6, High-Yield Summary and Question, Mastering the Microscopic Fortress.
We have covered a tremendous amount of ground today, class. Let's rapidly synthesize what we've learned to lock in that exam knowledge. First, phospholipids create the fundamental nonpolar barrier, while occasionally acting as internal chemical messengers. Second, cholesterol acts as your crucial fluidity thermostat and provides critical mechanical stability. Third, glycolipids and glycoproteins project outwards to form the glycocalyx, utilizing hydrogen bonds for stability. Fourth, those sugary antennae function as specific receptors for signaling, endocytosis, and adhesion. Fifth, they double as the unique antigenic flags for immune recognition. And finally, proteins are the heavy machinery, providing specific transport channels, acting as localized enzymes, and anchoring the cytoskeleton.
Now before we go, I want to leave you with a thought-provoking challenge. If a genetic mutation prevented a cell from successfully synthesizing carbohydrate chains, which specific membrane functions would fail first, and how would this impact the survival of the entire organism? Think about the flags, the antenna, and the cell structural stability. Take this question, connect the dots we've covered today, and you will truly have mastered the microscopic fortress. Thank you for learning with me today, and keep exploring.
Key terms for this topic are presented on the screen, classified according to different exam boards. These terms require dedicated focus during preparations as they are essential and should never be ignored. In addition, past paper analysis is included for each board and paper type. Be sure to review this carefully as it will greatly contribute to improving your preparations. Also, make an effort to understand the meaning and application of each keyword rather than memorizing them on their own. Regular practice using these terms in past paper questions will significantly enhance your accuracy and overall exam performance.
Please subscribe our channel to get more educational videos. Thank you.